Welcome, everybody. It's a pleasure today open the 2022 seminar series of the Nucleus for Scientific Initiation and Extension of the State of Para University Center. And it's a pleasure and a privilege to have today with us in this opening our speaker, Diego Szczupak. Diego currently is a postdoc at the Neurobiology Department of the Center for Neurosciences of the University of Pittsburgh, United States, and very gently accepted our invitation to be here. And Diego works mainly with brain mapping, using non-invasive techniques like magnetic resonance and its modalities, with very new techniques, maybe for us all. But the context of his talk will be very interesting, since many of our students and many of our professors are interested in neurosciences. So, Diego, thank you very much. Now the word is all yours. After Diego's lecture, we will open for question sessions. Okay, Diego, the word is yours.
Well, thank you very much for the kind words, Claudio. It's a pleasure to be here. And I'm very excited to talk to you guys about this work. And hopefully you guys will like it. You all see my screen?
Yes.
Perfect. So I'm going to talk today about a work that I have been doing for a long time that started in my PhD, and it's following me through the postdoc. And I put this provocative title, The Secret Pathways of the Brain, because I truly believe that we do not know yet enough about brain connectivity. And we do not understand how the brain functions. And I think the most iconic structure of connecting the brain is the corpus callosum, which is this nice little guy here in the middle that said that we need to work together. And he's saying this because he's the structure that connects both left and right hemispheres of the brain. And this is a work that is being a great collaboration between Brazil and in the US. It started off in the Federal University of Rio, and then in partnership with the IDOR Institute. And then I continued this work at the NIH under Afonso Silva's lab, and then I keep doing it here in the University of Pittsburgh. Okay, so the first thing I want to talk is, well, this is just something interesting that to get the conversation going about the corpus callosum. It's the story about the callosum dysgenesis. So to start with, there was in the, I think it was in the 50s, they started doing the surgeries to remove the corpus callosum, to cut the cross-section of the corpus callosum in patients that were epileptic resistant to medication. So the idea is that they would have an epileptic focus, and that would spread through the other hemisphere, and by cutting the corpus callosum, you would reduce the spread of it. And it was a very invasive surgery, but kind of helped a lot of the patients to not have such bad seizures. The interesting thing was that once you cut such a drastic bundle off the brain, you would imagine a lot of deficits, right? But interestingly enough, there were not many. These patients seemed to be fine at a first look. And it took the work of Roger Sperry and Gazanga to carefully study these patients and understand that, yes, there was something that they were missing. So, for instance, this is an object recognition test. As you can see here, the subject would touch with their right hand a scissor, and with their left hand they have to point it. So the idea is that if they touch the object with the right hand, the information will flow to the left hemisphere of the brain, which is the hemisphere that's responsible for the speaking. So the patient would be able to name the object and point it. On the other side, if the patient touches the scissor with the left hand, the information will flow to the right hand, and it will only be able to name it if the fibers from the corpus callosum can transfer the information back to the left hemisphere. And these patients that had the corpus callosum section, they could not name it. And there were other things, but this is the most iconic test. And now the fun begins. That right after that, there was an incidental discovery of a woman, I believe, that went to do an MRI and found out that she had no corpus callosum. She was just born without it, and she had no deficit. And then start to study and find out that all these patients that are born without the corpus callosum, or a good amount of them, they can name the objects, they can transfer information without the corpus callosum. How that is possible? Something must be happening in the brain. So over here, I show we call the dysgenesis of the corpus callosum. It's a broad spectrum, but roughly we can say that it can be an agenesis, where the patient has no corpus callosum over here, a hypoplasia, when it's a small corpus callosum over here, or a partial, where you have a part of the corpus callosum, usually the genome. And my lab in Brazil, it studied a lot these patients and used all these interesting techniques of diffusion MRI that can investigate the white matter of these brains and color code it in a way that you know where the fibers are going. So, for instance, the green shows in interposterior, the blue is dorsal ventral, and the red is lateral lateral. And you can see here in a T1 as well as in the DTI that you cannot see any corpus callosum, but you can see the cingulum bundle, which is a normal bundle, and you can see this big longitudinal bundle that's called a probes bundle. And the probes bundle was actually discovered in the 1900s, 1908, I think. But it was the first show that these patients could have some sort of plasticity in the brain that would connect it somehow. Or can like something is happening to the brain can change, the brain can adapt somehow. And then my PhD advisor, Fernanda Tovar, she did this diffusion imaging of these patients, and she found this abnormal tract. And I really want to point this out, as this is a heterotopic fiber that connects the frontal pole with the parietal occipital pole of the contralateral hemisphere. And this is something that's very odd, because we usually think of the corpus callosum as this homotopic structure connecting homologous areas of the cortex. And later on, this was shown in fetus with corpus callosum dysgenesis, and even on a T1 of a patient that had no corpus callosum. So, okay, these patients seem to connect it somehow, the hemispheres, and it seems to be through these cross connections that are heterotopic connections. So they were called the sigmoid bundle due to its shape, and it was presumed to be causing symptoms, since it should be something different from regular controls. Later on, they found that there are all these other bundles that connect the hemispheres, which I'll be happy to talk about later, but it's not really the point right now. The point starts in my PhD thesis, where we got these mice that are called the bulb C mice. It's a commercial strain that have defects of the corpus callosum. So you can see here one that has a full corpus callosum, a smaller one, and an absent one. And you can see here C57 black 6, which is the normal and the neurotypical strain. And they have a decent sized corpus callosum and very narrow distribution, compared to the bulb C that has all these wide range of distribution. And we redid the test at the NIH with 8 animals, and we also found this wide range of distribution. And then I started to study them with diffusion MRI. We had at the NIH a 14 Tesla magnet, and we're able to do 80 micron DTIs. And with that, I was able to reconstruct all the cortical connectivity. And the callosum connectivity, for instance, would be very different from animals that have the corpus callosum or animals that have a short corpus callosum. You can see all these areas that are not connecting. And you can even see that some areas are connecting through different paths. For instance, instead of going through the genome of the corpus callosum, the frontal connections are going through the hippocampus almost, or the hippocampus commissure. And we found the so-called sigmoid bundle in mice, which was very exciting. It's the first time it was discovered in mice. We did it at the same time as Linda Richards' lab with Tim Edwards, which is a great scientist from there as well. And it turns out that we found this sigmoid bundle. And I don't know if many of you are familiar with diffusion MRI. It has all these wonderful images, but sometimes it might lead to false positives. So we were like, okay, let's double check this. And we did it the way that we think it should be done. We used a virus, an adeno-associated virus, AAV9. It's a virus that you inject in the brain, and it will label the neuron. It will replicate along the neuron, but through the axon, but will not jump synapses. So it will be very specific to the connection that you're looking at. And you can see all of these are axial slices on a bulb C animal, and we injected in the frontal pole between the prelimbic and the anterior cingulate. And you can see that the fibers travel more posterior. They get closer to the midline. Some of them continue posterior, but some of them cross, and you can already see something here in the contralateral hippocampus. And you can even see the angle of the fibers going 45 degrees, and something a little messy, but you can see them clearly going 45 degrees and diving into the hippocampus. So we're like, okay, we're happy that we found the sigmoid bundle with histology as well. It is a real thing. And then the idea was, well, let's show that this was something that only happened in these animals. So we did it on the C57 animals. And for our surprise, we saw the same exact bundle. I wouldn't say exact. It was a little bit smaller, as you can see it over here. But it was there. It was clearly there. So we published this in your image, and then we're thinking, well, how much we actually know about the brain then? If the things that we thought it was pathological, they are real. They are common. Should we question other dogmas of the connectivity of the brain? And we did a connectomic analysis, where we do the same tractography here as I showed before, but we do it for the whole brain, and we ask from one region to another region through the whole brain what is connected. And this was an interesting thing. This was an analysis done by considering the whole population of C57 animals that we had and the whole bulb C. But instead of looking at the statistical difference, we asked the question, what is common to all the C57s and what is common to all bulb Cs? Meaning, is there like a core difference between these two strains? And as you guys can see, the binary difference is very big. Total connections, intrahemispheric, interhemispheric, and the ratio as well. And the weighted, and here's weighted by connectivity strength. It's also very clear, where the animals without corpus callosum have a more intrahemispheric connections, while the animals with corpus callosum have more interhemispheric connections, which all makes sense. But you guys can see here that all these connections here look very cross connections as well. So that started to make us curious. What is the corpus callosum really doing, and what is it actually made of? And then we did a different analysis. We decided to plot all the connections of C57 and then subtract the individual bulb C animals. And we realized that all these bulb C animals were different from one another. And a lot of them have a lot of differences in the frontal pole, which was expected due to the dysgenesis type and everything. But again, these cross connections seem to show up. And even here in the C57, they are very present and very numerous. So we did a different experiment now. There is this wonderful database called the Allen Brain Database of Connectivity Atlas, where they did all these injections with the similar tracer that I did. And we decided to look at them, trying to understand a little bit more the pattern. And what we found out was that there is a commissure that was not being described before. That's called, we named the thalamic commissure, as you can show it here. So the thalamic commissure is connecting the one side of the brain with the contralateral thalamus. You can see here for the orbital, as for the lateral orbital cortex, which is kind of like the dorsal prefrontal cortex of a human, and the MLP, which is like the M1 for a human. And you can see that they connect through this new commissure. And then we're a little bit skeptical now about what we knew. So different commissures, different everything. And now this talk actually starts about this work that we have been doing. We just submitted, and we're very excited about it. And I hope you guys will be as well. So this is an injection in the frontal pole of a mouse. This is a dorsal view of the brain. You can see in green all the projections that this region has. So you would see that over here has a large projection to one side and a little wide projection to the other side as well. What would you normally expect from the corpus callosum, what is being very described in the literature and is a dogma that we have, is that it would connect the homotopic region right here. And what we can see is that although it's mirrored, it is connecting all these heterotopic spots. With diffusion MRI, it would be something like this. We would be expecting the blue, but we are also seeing this red one. So we went back to the database, to the Allen Brain database, and we chose 4 different spots throughout the brain. So one is the orbitolateral, and you can see some heterotopic connections, not many, but some. Then we saw the somesthetic, the primary somatosensory cortex, where you can see more heterotopic connections. So this would be the homotopic, and these are all heterotopic connections. And then the visual cortex, and the same pattern shows. And one that got me very excited and very impressed was the ectorinal cortex, which has projections all over the cortex, including the contralateral cortex. And here I put some videos just to show these connections in 3D, so you guys can see how spread out these connections are along the brain. And it just goes to show that the corpus callosum is actually doing a lot of different stuff than what we thought. There are a lot of heterotopic connections. So we decided to actually quantify that. As I showed here before, we have an atlas from the resource of the Allen Brain database, and we have the projections. So we calculated. We calculated what we call the heterotopicity index for each region. And the heterotopicity index means that how many fibers are homotopic, how many fibers are heterotopic divided by how many fibers are interhemispheric from that region, that originate in that region. And what we found out is that most of the brain is actually heterotopic, with some regions like the ectorhinal cortex being very heterotopic. But that in numbers means that from the whole brain, around 20% of the connections are interhemispheric connections. From those interhemispheric connections, 2/3 are heterotopic connections. So the corpus callosum that we always thought that were connecting those regions, those homotopic regions, is actually connecting a lot more. Two-thirds of those connections are not thoroughly investigated yet. And they go for almost 10% of the connections of the brain. Furthermore, the idea that we saw before about these heterotopic bundles uh, being causing symptoms and being something exclusively from pathologies, it's not true either. They are normal in the brain. They are abundant in the brain. When we don't have the other bundles, is that's easier to notice them. But I'm getting ahead of myself. I'll go back to that later. Then what we did was since we had this discovery, we decided to do the fusion MRI now, and to map all of these same connections in the mouse, but not only in the mouse. We work here with marmosets, which is a non-human primate, natural from Brazil. I'm sure that you guys know it very well, and from humans as well. And we found out that, again, roughly two-thirds, a little bit more to three-quarters over here, of the connections of the brain are heterotopic. And we can see that they are in very distinct patches and they are matching patterns here and here. And this is similarly conserved through evolution, through rodents, through marmosets, through humans. And more than that, we decided to map now these heterotopic connections within the corpus callosum to discover that there is actually a gradient of the corpus callosum, where the heterotopic connections are more in the middle of the corpus callosum, while the homotopic are more in the periphery of the corpus callosum, which reveals a gradient that probably have an not evolutionary, developmental origin. And finally, what we did was that we got we did the same analysis as I showed before with the connectomics, but we got the what we call the normal connectivity, the full network of the connectivity, so all the connections of the brain, and we compared with the same brain with the what we call the non-heterotopic. So we only removed the heterotopic connections. And by comparing them, we see that these heterotopic connections that were removed have a great importance and a great significance to the efficiency of the brain, to the small worldliness, which is a pattern related to the efficiency of the organization of networks in general, to the hierarchy, and to their sortativity in mice, marmosets, and humans. And we think that these results actually are of broad interest because it not only affects the research of people who work with dysgenesis of the corpus callosum and things like that, but actually it kind of makes us review the idea of development of these fibers, because we usually think about them being homotopic, also because we understand that the formation of this structure is by fasciculation, which is when one fiber kind of merges with another one and kind of goes together, like sticks to it and uses the other one as a rail and reaching the same targets. But this shows that that might not be the case, or at least it's not the case for two-thirds of it. And it kind of puts more questions than it answers, but it opens an avenue of understanding how the brain is actually connected, how one area can affect the other hemisphere, or actually the whole brain, if you think about it. And what are these connections doing? And this is also vital for people who are doing modeling, as we just shown here with the graphs, and people who are doing electrophysiology and assays of function MRI and task, because we usually think about a hemisphere as a mirror of the other one. And we usually think about it as well as kind of like a closed system where one area connects to the other, but not really. What we're showing is that everything is connected at the same time. Not saying that all of them are driving the brain at the same time. Possibly most of these connections are modulators, but still there is a tight connection between different areas of the brain, and there is the system is much more complicated and much more intricate than what we previously thought. So that was it. And thank you very much for listening to me. And I just want to present you guys about for our lab over here in Pittsburgh. And this is Afonso Silva, my PI right now. This is our scanner that got here uh, late last year, and we're very happy with the images we're acquiring right now. This is Roberto Lente. I'm sure most of you guys already know him from FRJ, Fernanda Tovar from my door. And this is Pamela Menezes, who has been incredible help, and she's a PhD student and has been collaborating with me in this work. Well, thank you very much. I hope I was on time. I guess it actually was a little fast.
Sure. Thank you very much for your lecture. Your title is as much provocative as your results, and
I think that's a compliment.
yes, I'm a bit amazed about your findings. There is one thing that called my attention more prominently. You showed that different regions in that heterotopic connections made through the callosum, they show different spatial distribution of networks. And when we do network analysis, we consider some parameters like randomness, heterogeneity, modularity, independence of the type of the parameter and the combination of such parameters. From the network emerge different functions. And I'm thinking about functional MRI in combination of a network analysis of this spatial distribution, what could be revealed in such analysis.
No, that's a very good question.
You are thinking about something in this way?
Yes, that's a very good question. So for sure, I do believe that these connections are letting the brain to be able to process more information and handle more. Well, the same way that a city that's more connected with roads can handle different types of rush hours and events and things like that. I do believe that more interconnected brain regions can handle more information and can process things in a different way and better way. Um, about functional MRI, it gets tricky. We have tried this because the issue is that you don't see monosynaptical anymore. So you don't know exactly what's mirrored, what it is, where it is, and we're trying something here now. And I want to get around of that. That's manganese tracing. Manganese, it's a calcium analog.
Yes.
And it also shows up in the MRI. So if you inject manganese in one point, it will trace to another one, but it will also jump synapses, so we can actually follow it. So that will be very interesting for this type of stuff. And you also ask about if I believe that the emerging properties would be different, right? And we have been working on different species now, but we have also been working with different diseases. And I don't want to say anything because the results are not set in stone yet, but it seems very interesting that yes, we can correlate this somehow, the heterotopicity index with different cognitive measurements.
I can imagine that, for example, in a stroke, and some patients have greater losses than others, and the impact of such strokes on the distribution of such heterotopic and nomotopic proportions of connections through the callosum certainly impairs the randomness of the previous normal organization of this network, the heterogeneity and modularity of the distribution of such connections. I can imagine that distribution of synapses through these connections make the modulus, for example, and the distribution of modulus determine the heterogeneity of these connections. and therefore the randomness of the network is well defined by these previous parameters, and the stroke on it disturbed this organization, and a new organization comes on, and the resulting emergent function should be proportionally disturbed. So it can be a new way to see the disease. It's a new way to see how the prognostic of the patient can be followed, and it's very promising. Your results are amazing.
Thank you. I really appreciate it.
I'm very happy to see this in our first seminar in this new series of this year, 2022. Let me see if we have some questions from someone here. And applauses, a lot of applauses, no questions. Diego, thank you very, very much for the acceptance of our invitation. Gentle from your part. I know you are very busy. And so we can end this session now. Thank you very much.
Well, the pleasure is all mine. Thank you very much for the invitation.